Okay, we are starting out a topic on Mendelian genetics today. And so, Mendelian genetics as a phrase comes from this man right here, Gregor Mendel. And so, if you know anything about Mendel, some of these diagrams might make sense to you. I'm actually going to start us with a little bit of a video that gives you a short biography into who he is and what he did.
Monk teacher and biologist, Gregor Mendel is known today as the father of modern genetics.
Gregor Mendel's experiments on pea plants were fundamental in establishing the field of modern genetics.
Born Johann Mendel on July 22, 1822, in what was Austria and is now the Czech Republic, Mendel excelled at school and became an Augustinian monk to further his education. He was given the name Gregor when he entered the monastery.
The monastery sent him to the university in Vienna, and that gave him the education in science, math, chemistry, and botany that he then brought back to use in his teaching.
Mendel accepted a teaching position at the monastery in Brno after completing his university studies. It was during this period he began his famous pea plant experiments.
Before Mendel, people thought you would equally inherit the traits of parent A and parent B in equal proportions.
Mendel experimented with pea plants and found that instead there were dominant and recessive traits in all organisms that were passed on statistically based upon the mixing of genes between the two parents.
This theory is now known as the law of segregation. Mendel also formulated the law of independent assortment.
The law of independent assortment addressed the fact that there are a large number of traits. These traits are passed on randomly and are independent of the other traits and of the traits of the two different parents.
The ability to explain hereditary diseases and to engineer plants with desired properties are all consequences of the fundamental theory founded by Mendel.
During his life, Gregor Mendel's research was disregarded by almost everyone. It wasn't until the early 1900s, after his death, that Mendel's work was appreciated and now serves as the basis for genetics and heredity.
In his later years, Mendel became the abbot of his monastery and spent less time with his scientific endeavors. Gregor Mendel died in 1884 at the age of 62.
Gregor Mendel's work laid the foundation for modern genetics, for modern gene therapy, and is the basis for some of the most exciting discoveries in medicine currently and in the near future.
All right, so you can see, let's go back to, here we go. He was an Austrian monk, and I give you the date range just so you have some perspective on how long ago he lived, mid to late 1800s. As the video said, what he discovered and sort of the experiments that he was able to run in his time were not terribly significant, not accepted widely by the scientific community. Although now today we know that what he discovered and kind of what he thought lays the basis for our understanding in genetics. And so he is considered the father of genetics. So let's talk about his experiments. So we know that he worked with pea plants, and that is probably for two reasons. Number one, the monks gardened to produce food for themselves. They were pretty self-sustaining. And so, yes, he was an avid gardener. He was a scientist. He enjoyed observing and collecting data, but also he grew their food. Pea plants in particular, if you look down here, they come with several different kinds of very easily visible traits. So when it comes to the seeds, it's very easy to see if the seeds in a pea pod are round or if they are wrinkled. It's very easy to see things like flower color on the pea plant. Are they white or are they purple? It's very easy to see the color of the pea pod. Is it green? Is it yellow? And so it was easy to work with pea plant traits because they were easily observable. Now, you have to understand that previous theory at the time of inheritance was something called the blended theory or the blending theory. And that's the idea that you are a blending of traits from both of your parents. Meaning, if I mix a red can of paint with a white can of paint, I get sort of this brand new color called pink that is a blend, a mix of the previous two colors. And so what Gregor Mendel was discovering through his experiments completely turned that previous theory of blending on its head. And I'll explain why in just a second. So one of his more famous experiments, he crossed a pea plant that had yellow peas with a green pea plant. And so blending theory would tell you that the offspring from this yellow and green would produce a blended color between the two. But in fact, what he discovered is that the offspring were all yellow. Now that's kind of interesting, number one, because there's no blending of colors here. And number two, where did the green color go? So this was an unusual finding. Oh, not where did white go? Where did the green color go in the actual pea pods? And so ultimately, he continued to run experiments like this. And ultimately, where this is leading him is the idea that actually inheritance is not a blending of traits at all, but rather there are different kinds of types for any given trait, and one can actually dominate over the other. And so we understand this now to be what is called complete dominance inheritance. When you have a dominant allele, and I'll explain what that means in just a second, it completely overshadows a recessive. And so that was a really big deal at the time because it showed that this whole blending thing, it's not really a thing. In terms of what he is discovering, it's not quite a thing, but rather one of those colors was actually dominating the other. If we keep going into the next section, we're going to touch on some vocab words that are associated with Mendel, his experimentation, and his discovery. So the first couple of vocab words that you need to understand come from this diagram right here. So these letters off to the side, P, F1, and F2, these are all indicators of generations. So the P generation, that's pretty easy. That's the parent generation. And the F1 generation is pretty easy to understand as well. So these are the offspring from the two parents that have mated together. So these two pigeons, these mom and dad pigeons, produce these possible offspring, and that is called the F1 generation. Now, if we go down another generation to F2, notice in the diagram what produces an F2 offspring interbreeding two F1 pigeons together. So when we talk about an F2 generation, it's not just the next generation down, it's actually interbreeding two of the F1s together. So keep that in mind as you read diagrams, P, F1, F2. There's also an F3 and F4, and those generations are very similar to what we see in the F2. It takes an interbreeding scenario to produce two F2s breeding together, give us an F3, and on it goes. Now, when we talk about traits, we're talking about physical characteristics, and then we're also discussing something called alleles. And so alleles are the fancy word. At the time, Mendel did not coin this term. He called them factors. But alleles are different variations of a particular trait. So for example, if you check out these chromosomes down here, here is the A trait, whatever it codes for. And one version of the A trait, or one possible allele for the A trait, is a dominant allele, capital A. The other possible version of the trait is recessive, indicated with a lowercase letter. And so when we start to talk about the different combination of alleles that one individual could possibly inherit by their parents, we're talking about that person or individual's genotype. And so here's what I mean by that. If individual has two dominant alleles, that individual is called a homozygous dominant genotype. They have a homozygous dominant genotype. If they have two recessive alleles, one from mom and one from dad, one from the sperm, one from the egg, then they are considered homozygous recessive. And if they end up with one of each, maybe a dominant allele came from dad in the sperm and a recessive allele came from mom in the egg, they are considered a heterozygote or a heterozygous genotype. So genotypes are the combination of alleles that have been inherited from parents. Now, there's another type called a phenotype. Phenotypes are the physical characteristics that the individuals are actually displaying. So what do they actually look like? And so, for example, over here in this diagram with these flamingos, the genotype of this first flamingo is big A, which is homozygous dominant. The phenotype is red. This flamingo or bird is kind of a reddish pink. This one, the genotype is big A, little a, or heterozygous. The phenotype, what they actually look like, again, is red or kind of a pink. This bottom one, the genotype is homozygous recessive, little a, but the phenotype, what it actually looks like, is blue. Now, there's another term that Mendel's experiments kind of give us, and that is the term pure breeding, or we sometimes see it as true breeding. They can mean similar. This is a homozygous individual that can really only contribute one allele type. For example, if you're homozygous dominant, you only have dominant alleles to give. If you are homozygous recessive, you only have recessive alleles to give. So you are true breeding or pure breeding because you can really only contribute one allele type. So there's a good example. Homozygous dominant, big P, maybe you produce a purple flower. Homozygous recessive, little p, you produce a white flower. Some of you might be going, why little p for white? When we're talking about a trait, in this case, flower color, you typically, whatever the dominant trait is, purple in this case, sets what letters you use. So in flower color, the P's are representing the color. If the P's are capital, it's purple. If the P's are lowercase, it is white. So why are true breeding, pure breeding organisms even important to talk about? Because there's something called a test cross that Mendel introduced us to, but that often is talked about in a lot of question sets and experiments. So a test cross is what you would do if you have an organism that is the dominant phenotype, but you don't know what their genotype is, meaning you don't know if they're homozygous dominant or heterozygous. All you know is that they show the dominant phenotype. And so if you are trying to figure out which of these two your dominant phenotype organism is, you would cross it with a true breeding recessive. So here's what that would look like. Let's say we have this black sheep. We know that black is the dominant phenotype, but we don't know if this black sheep is black because it's a homozygous dominant or it's a heterozygous. So to figure that out, we would cross it with a pure breeding recessive, this white sheep over here, which we know only has little b's, recessive alleles, to contribute. And so we would look at the offspring, and here's what we would look for. If all of the offspring came out black, that is an indication that the parent that was the unknown is actually a homozygous dominant or a homozygous dominant genotype, big B. That would produce, even when mixed with the white recessive, all of the babies would still be black. If this organism were a heterozygote, when you crossed it with a recessive, you would get some black sheep, but you would also get some white ones, because that recessive allele, big B, little b right here, that recessive allele would combine with a recessive allele from the white sheep to give some white babies. So a test cross is when we don't know actually the genotype of a particular dominant individual. We just know what they look like. We know their phenotype, but we don't know their genotype. And so we test cross it with a true breeding recessive. If the offspring shows recessive, then the dominant that was in question was actually a heterozygote. It had a recessive allele kind of hidden in its genotype. All right, so here's a quick check for you. First, look at this diagram. What is the genotype or genotypes? And then what is the phenotype or phenotypes? So pause for a second. If I ask you genotypes, you're giving me these letters down here. So the genotypes in this diagram are big A, which is homozygous dominant, big A, which is a heterozygote, and little a, which is a homozygous recessive. The phenotypes, the physical characteristics that I see, are purple and white flowers. So then check number two, a genotype always determines a phenotype. True or false? Does a genotype always determine a phenotype? Does the letter set always determine the color or the physical characteristic? Yes, it does. That's a true statement. Number three, flip it. A phenotype always determines a genotype. What do you think about that? That one is false, because a purple, in this diagram especially, a purple flower, a phenotype of purple could be either this genotype or this genotype. So it doesn't exactly work the opposite way around. All right, next section, we have Mendel's 3 laws. Now, these are pretty quick because actually we've already seen these at play in meiosis. So he gets credit for 3 laws. The first law is called the law of segregation, and that means that during meiosis, the homologous chromosomes will segregate or separate into different cells, and this happens at anaphase 1. That ultimately creates gametes with just one of each homolog in it. And we have seen that in meiosis. His second law is the law of independent assortment. That should ring some bells. That is saying that when those homologs line up at metaphase 1, they do so independent of any other pair's arrangement in the cell. And we know that that gives us a whole lot of variation in the offspring. And the third law that is attributed to Mendel is called the law of dominance. And that means that if an individual possesses one dominant allele for a trait, that's enough that they're going to express the dominant phenotype. So all it takes is one big B for the sheep to look dominant in color or black in color, using the diagram from a couple of slides ago. So Mendel gets credit for these 3 laws, what he discovered as a part of his kind of experimentation. Now, the last thing that I want to show you so that we can all be at a place of practicing together in class is a Punnett square that is called a monohybrid Punnett square. Now, you've probably seen these Punnett squares, these little boxes or crosses. What I'm getting ready to show you, you've probably done before, which is why I'm kind of zipping along. So a monohybrid cross, mono as a prefix means one. This is a one trait cross. You are only looking at one particular trait in this cross. And for example, it looks like the trait that we're looking at over here in this cross is the color of the flower. Now, this big box, it's really just a model. And so it models the possibility or probabilities that 2 parents can make when it comes to their offspring, if they're going to sexually reproduce with each other. And so if we look at this model on one axis, which happens to be this axis. Here's the female. On one axis, you have the possible, so mom's genotype for this purple flower was big B, little b. And so when she goes through meiosis, her B's, her homologous chromosomes with the B's on them, they're going to line up, and then they're going to go through meiosis and they're going to separate. And she's going to produce some eggs that have the dominant allele in it, and she's going to produce other eggs that have the recessive allele in it. So we see mom's information on one axis, and then we see dad's information on the other axis. So dad was also, with a purple flower, was a heterozygote for that purple color. So when his homologous chromosomes lined up and he created, and they split and he made sperm, some of his sperm had a big B for the allele of flower color, and other sperm had a recessive allele. And so then we allow these sperm and egg to meet and fertilize and create a baby. And so this box right here, you are looking at a possible offspring that would occur if mom's egg with her dominant B was fertilized by dad's sperm with his dominant B. That would make this baby right here that would have purple flowers and a genotype of big B, big B. Let's look at this one. This is a possible baby that these two parents could have together if the egg with mom's recessive allele in it was fertilized by dad's sperm with his recessive allele in it. They're going to make a baby whose genotype is homozygous recessive and produces that fully recessive trait, or that white flower color. So the crossing, really, it's a skill, and you probably have learned it before, and it's probably quite simple to you. But what I want you to know right now for AP Bio is that the cross is great, but what the cross is modeling is just the possibilities that could occur between a male and a female when they reproduce sexually. What kind of babies could they create for that particular trait? In this case, it's flower color. Now, once we actually do the cross, we give a genotypic ratio of our cross. And so a genotypic ratio means how many of each genotype. So if I look at the offspring, I have one that's homozygous dominant, I have two that are heterozygotes, and I have one that is a homozygous recessive. So that's a 1 to 2 to 1 ratio, and make sure you put your key at the bottom so that we know what this one refers to, and this two refers to, and this one refers to. We also give a phenotypic ratio, which in this case is 3 to 1, which means phenotype, physical characteristic. There are 3 purple flowers and there is 1 white flower in the cross. So make sure again that you give yourself a key under your numbers so we know what they relate to. Now this is basic and simple, and we'll practice this in class, and then we're going to move on to the more complex, because this is probably what you learned last year, and we definitely need to cruise into new AP Bio type stuff. But when you're done here, you are done for the moment.